Hi everyone, welcome to the video. This is going to focus primarily on the topics in eukaryotic gene regulation that we didn't get to in class, and that we'll review, but I didn't want to show for the first time. So what you're going to need in order to kind of follow along is the worksheet that I gave you in class that shows all the major images for eukaryotic regulation that you need to know, so that you can pause and take notes as you see things that are important and valuable to you. So I'm not going to go through the ones we saw before. We should be familiar with chromosome and chromatin structure by now. Histone proteins. Remember, we saw the difference between euchromatin and heterochromatin by how tightly wound the DNA was around histone proteins, and that can control access to promoter regions. We looked at this idea of a nucleosome, or a bead of histones, and this idea that we could modify histones and DNA to change the relationship that they had with each other. So by acetylating histones, we see a looser association between DNA and histone proteins. And by methylating DNA, the opposite of acetylation in this case, we are impeding transcription and reducing the chance that transcription will occur. So we are essentially associating histones and DNA more tightly with one another. We saw this that showed that acetylation would loosen, or in this case, deacetylation, so the removal of acetyl groups, but that is where we're at here. So where we kind of left off in class was this idea of transcription factors and how they assemble in what we call the transcription initiation complex. And a word that we want to keep with us is this idea of control elements. Now, control elements are DNA sequences, and more importantly, these are not parts of the gene themselves, but they are DNA sequences that have relationships with particular proteins, and in doing so help to control transcription. So we have two different types of control elements. We have proximal, which we'll look at first. Proximal are the ones that are control elements like the promoter sequence, for example, that are near the gene, usually just upstream of the gene, meaning that they are, to the left, they're before the gene. And so these are parts of the promoter, and they will typically encourage regulatory proteins such as transcription factors that can do one of two things. They can either enhance transcription or they can inhibit transcription, just like we saw with the operators in operons. There's now this second class of of control elements called distal control elements. And these are DNA sequences that are typically removed quite distally or distantly from the gene. They can either be silencers and stop genes from being transcribed, or they can be enhancers that further promote transcription, and they will bind our transcription factors as well. So let's take a look at what we're talking about here. So we'll start with our orientation here. We'll start with the promoter sequence. These are proximal control elements, meaning that they are close to the gene, and they bind and have sequences that bind to these transcription factors and mediator proteins. Now, far away, all the way on the left-hand side of this diagram, is something called an enhancer region. And you can see that this is quite removed. It doesn't have to be this quote-unquote close. It can be much farther away, much more distal. But these particular sequences here, these sequences that are highlighted in yellow, will bind to specific proteins called activators and help to kind of create this complex of molecules. And so as this enhancer region bends and begins to interact with this larger complex of proteins, we have general transcription factors, we have mediator proteins, all converging on the promoter sequence, delivering RNA polymerase to its promoter and initiating transcription. And it's not until all of these things come into contact with the promoter region that RNA transcription can take place and polymerase can do its job. And so in this transcription initiation complex, in the absence of any of these proteins, the rate of transcription will go down, and the effectiveness of transcription will decrease. Now we're only looking right now at the influence of activators. There are also specific proteins that could inhibit. We'll just draw it with an eye. It's a little tedious to draw with the mouse. But there are inhibitors that will get in the way of this as well and prevent transcription. So this is just another look, but a similar concept where this enhancer region will bind to these activators in green and help make sure that this complex of transcription factors actually make it to the right site at the promoter and move forward. Now, to give you an idea of why this is important and why enhancer regions can help further control where in the body certain transcription takes place, let's look at two examples. And so we're going to look at two genes here, an albumin gene, which is something that is necessary in the liver, and crystalline, that is important in the production of lens cells, so the creation of the lens in the eye. Now, because our DNA is omnipresent and we have copies of all of our genes in all of our cells, it is not the presence of a gene that determines where and when it is transcribed, but the presence of certain activators that interact with these enhancer regions. So if we look first at the left-hand side, and we look at our liver cell, we'll see in the liver cell nucleus there are a few different activators available. There is a red one, there is a gray one, there is a yellow one. And if we look at the albumin gene on top, all three pieces of this enhancer region have an activator protein attached to them. And in doing so, this allows the transcription initiation complex to form and for albumin gene to be expressed. In the crystalline side of things, even though there is one activator available in this gray activator, the other two enhancer portions, these distal control elements, are absent of protein, and because of that, the crystalline gene is not expressed, and the transcription initiation complex is not achieved. If we go to the right-hand side and look at the nucleus of a lens cell in the eye, we'll see that there are different activators available. There is still the gray one that is necessary for both genes, but now we have a pink protein and we have an orange protein available. And these are proteins produced from other genes or have come to cells through alternate means, but they are available in the nucleus and control transcription. And so the albumin enhancer does not have all of the necessary proteins attached to distal control elements for expression, but the crystalline gene does. And because of the regulatory proteins that are in the nucleus, this crystalline gene is expressed instead. And so we can see that in the presence of the right activators or in the lack of presence of certain inhibitors, we have transcription occur. So that kind of concludes the control of transcription. Well, we have here a few different mechanisms for controlling gene expression after transcription has taken place. So we call this post-transcriptional control. And it can include four different things that we're going to look at here. This alternative RNA splicing, mRNA degradation, control of translation, and protein degradation. Alternative RNA splicing, something that we may be familiar with. And essentially what it does is it allows our spliceosomes to control which exons in a strand of premature RNA end up in the final process. So if we look at our DNA strand at the top, you'll see that there are 1, 2, 3, 4, 5 different exons present that transcribes into 5 different RNA exons. But there are different combinations of exons that can lead to the creation of different proteins. In this first example, all 5 exons are included, and it makes for a much more complex protein A. In our second example, exon 3 is removed or not included. It's almost included as incorporated as an intron and removed. And in doing so, there is a subunit, this bottom loop, that is not included in protein B. Likewise, in our third one on the right, exon 4 is not included, and so we are missing this top loop that is present in protein A and B. And so by RNA splicing, or this alternative splicing, instead of having just one protein outcome, we can have multiple different proteins created from just one gene. And so instead of having just 20,000 genes in our genome, that can be translated and manipulated into hundreds of thousands of different genes, depending on how these exon sequences are spliced together. mRNA degradation is a pretty cool one. It's essentially, once our mRNA is created, there are small RNAs that can be used to destroy messenger RNA and stop them from being translated. So it's a little bit complicated. I hope I don't run out of time here in this recording. It's life. But essentially, we have microRNAs and we have silent interfering RNAs. And what they will do is these small RNAs will be chopped up into single strands, and they will marry themselves with a protein complex. The type of protein that is made is an Argonaute protein. That is the first one that it associates with. It's going to be a challenge to spell this, but it is Argonaute. And what this protein does is it connects with this microRNA here and helps form a complex, a larger complex called RISC. It is the RNA-induced silencing complex, and its job is to connect with a strand of RNA, this red RNA that's being translated, and essentially degrade and chop that strand. So the microRNA that's here is like a guide that takes this RISC complex to a specific messenger RNA in the cytoplasm, and the RISC protein cleaves the mRNA, breaks it down, and stops it from relaying its message. The final thing that we'll look at is what happens when proteins are misfolded or they incur some sort of damage and they need to be destroyed. If we look at this guide here, we have the central dogma at the beginning, where we have our DNA becomes RNA becomes protein, and there's two different outcomes. It can either be misfolded and jump right to this stage here, or it can be folded properly and damaged. Once it's identified as misfolded, it will be tagged by enzymes with these little circles here called ubiquitin. And what ubiquitin is it's essentially a tag that attracts these larger protein digesters called proteasomes. And these proteasomes will act as a little shredder that interacts with these ubiquitin tags and shreds it back down into its amino acids. So it's essentially digesting fully made proteins back into their amino acid components.